Good. Okay, so I want to welcome our speaker today, Dr. Koty Sharp. Dr. Sharp is an associate professor of biology and marine biology at Roger Williams University. At Roger Williams, she is also part of the team that comprises the Center for Economic and Environmental Development. Koty received her BA in biology at Mount Holyoke College and her PhD from Scripps Institution of Oceanography at UC San Diego. From 2006 to 2008, she was a Marine Science Network postdoctoral fellow at the Smithsonian Marine Station in Fort Pierce, Florida. And then from 2008 to 2011, she was a Goulet postdoctoral fellow at Ocean Genome Legacy at New England Biolabs in Ipswich, Mass. Before joining Roger Williams in 2015 or 2016?
2015, yeah.
Yeah, Koty was on the faculty at Eckerd College in biology and marine sciences. A little bit more about Koty, sort of personally. She was born and raised in coastal Delaware and is good family friends with the owner of Dogfish Head Brewing Company.
Come on.
So those of you guys who like that beer, hit Koty up. She was born and raised in coastal Delaware, but she has lived in California, Florida, Massachusetts, and Rhode Island. Her research has taken her to the Republic of Palau, Panama, Belize, California, and the Florida Keys. When she is not delivering research-rich coursework in her Roger Williams undergraduates or mentoring her research team in the lab, she is most likely swimming, kayaking, gardening, chasing squirrels and chipmunks, or spending time with her husband and two sons. So, Koty, thank you. Welcome.
Thank you, Jim. Thanks for the introduction, and thank you for the opportunity to talk with everybody today. A big thank you to Jim for coordinating these kinds of events summer after summer here for the SURF program. It's really I think it's I like to think that this is a really great opportunity for the SURF community, especially the undergrads, to just get a sense of and get experience with going to weekly seminars and forming this community of researchers across the state of Rhode Island. And one of the things I'll actually talk about today in my talk is how important community and collaboration is to research. So we really do, as the scientists and the researchers and the mentors, appreciate the work of the EPSCoR program, especially Jim, of all the corralling and herding and coordinating that you do for our summer experiences. So thank you.
Thank you. It's my pleasure.
So what I'm going to be telling you about today is some of the research that goes on in my lab here at Roger Williams, and I've divided it into a few stories that are sort of some microbial vignettes, I guess is the best way to think of them. We work with the coral that you can see here in the background. It's called Astrangia poculata. It is now officially the Rhode Island state coral, and I'll talk to you a little bit at the end of this talk about why that's important. But we use this coral to learn more about coral microbiomes and more broadly animal microbiomes and their dynamics, how they respond to global change, to changing climates, to changing temperatures. And because we know that microbiomes are so important for the health of their animal hosts. And so understanding how microbiomes shift and respond in the face of climate change, we think is very important for understanding and predicting the survival of animals in our future planet. And so I also want to acknowledge that INBRI and the CAIM program have been exceptionally supportive of my program over the years, getting everything up and going as I've been working here at Roger Williams. And we're gladly a part of the Microbiome Consortium here in Rhode Island. And I have to, especially because we're right in the middle of season 4, put an extra plug in for former INBRI fellow, actually, Aaron Borby's beautiful design of the Astrangia Things logo, which continues to be a sign of the work that the Astrangia research community does. And also, I will tell you that now that I've seen and worked with and given stickers out with this logo on it for years and years, when I talk to friends that are not in science and I say, oh, did you see the new episode? I usually make a misstep and I say, do you see the new Astrangia Things episode? So for those of you who are fans of the series, just know that my lab is, for better or worse, also very emotionally invested in that for all kinds of reasons. So Anyway, we do study Astrangia poculata, the northern star coral, which is, as I said, this coral that you're seeing here on the slide. What I want to talk to you about today is give you a little bit of background about why we think it's so important to study animal microbiomes, and thinking about animals as really not just the host animal itself, but a meta-organism that's facing climate change, and then tell you a little bit about what makes Astrangia poculata, this northern star coral, so unique, and why we use it to ask very specific questions about symbiosis. And then I'll tell you, like I said, a few microbial vignettes about what we've been learning about how microbiomes function in Astrangia and how they change in Astrangia, and end with some stories about establishing Astrangia as a useful experimental system for, like I said, for very specific questions about corals and microbiomes. And so to start off, like I said, all animals really are actually what we can think of as meta-organisms. So they contain they really represent multipartite or multi-domain symbioses. So there's the animal host as shown here in this figure. And in this case, we're talking about a coral. This figure is referring to, it was made for tropical corals. And in tropical corals, we know that there are endolithic algae. And so I'm sort of starting on the figure at about 2:30, 3 o'clock here. They're endolithic algae that live inside of the rock skeleton of coral animals. And then there also are archaea, and there are bacteria that live in the coral tissues, in the skeleton, probably in the surface mucus layer of corals. And then we also know that there are abundant viruses and fungi in those animals as well. The most well-known or well-described symbiosis within corals is an obligate symbiosis in tropical corals with the family of microorganisms known as Symbiodiniaceae. And that's what's shown here down at the bottom. They're microscopic algae that harness light, via photosynthesis. They make sugars and they feed their animal host with those photosynthetic products. And so that's an obligate symbiosis of tropical corals, which live in very carbon poor and nutrient poor waters. If they don't have the Symbiodiniaceae, they will run out of food. And so that has, of course, gained a lot of attention in the past couple of decades as we're seeing more and more outbreaks of what we know as coral bleaching, which is basically the loss of those symbiotic algae from the corals. And corals sometimes can survive that bleaching if they can reacquire symbionts, but oftentimes they don't. And if they don't reacquire new Symbiodiniaceae in their tissues, they can basically starve to death and die from that. One of the main triggers of coral bleaching is, of course, increased sea surface temperature. And so as we're facing global climate change, we're seeing massive die-offs of corals due to coral bleaching. That symbiosis, like I said, because of the phenomenon of coral bleaching, has been very well described. We're, of course, there's still, excuse me, there's still so much to learn about it. And and much work is still focused. You know what, I'm going to take just a sec here. Okay, sorry. Okay, sorry. I'll try that again. So coral bleaching is this phenomenon that has resulted in many scientists working really hard to understand that obligate association between Symbiodiniaceae and the coral host. But the bigger story that's extremely complicated and has captivated a lot of coral researchers for the past, I'd say, a couple decades now, is that there's a really complex but what appears to be specific microbiome associated with a lot of tropical corals. And they're in the skeleton, they're in the tissue, they're in the surface mucus layer. What we know now is that these microbiomes are very important for cycling nutrients, they're important for cycling carbon and waste. And many bacteria that live on the surface mucus layer really represent the first line of defense for the animal host. So they produce bioactive compounds that defend the coral host against potential pathogens from the seawater. So microbiomes play a major role in the health of tropical corals. Now, one question that I have been really interested in ever since I started working on corals during my postdoc is how are these different partners connected to each other and how do they potentially regulate one another? So we know that in tropical corals, this obligate symbiosis with Symbiodiniaceae is so critical for their survival. Are there ways that the bacteria and archaea, which I'll refer to as the microbiome, can impact the Symbiodiniaceae? Do the Symbiodiniaceae impact what microbes in the microbiome are present or are able to flourish in the corals? Those kinds of relationships are extremely hard to tease apart because, as I mentioned, when corals don't have Symbiodiniaceae, it's typically because they've been stressed, for lack of a better word, exposed to high light or high UV or high temperature um, stresses, and then they bleach. And then we don't know if the response of the microbiome is due to the loss of Symbiodiniaceae or due to the presence of other environmental stressors. And so this has been a question that I've been really interested in answering for a long time. And it's very hard to do this with tropical corals, especially in the lab and with experimental manipulation. So what's great is that there's this temperate coral that can be used to address these specific questions. And the reason why is that Astrangia poculata, which you're seeing here on the in these pictures, exists in the wild, both with high densities of those Symbiodiniaceae and also with extremely low densities. And so hopefully on the picture on the left, what you might be able to see is that there are coral colonies growing on a granite wall. This is a wall from Fort Wetherill from Jamestown, Rhode Island. And we see this occurring naturally where there are some brown corals and some white corals. And so visually to our eye, we can see the difference in symbiont state just by looking at the corals. And so what we find is that typically there are some that are what we call symbiotic, and they're sort of a range of brown from sort of a light tan to a dark, dark chocolate brown. There's what we call aposymbiotic, which are not completely free of those Symbiodiniaceae, but have very, very low densities. And then there are a lot of colonies, I would say actually the majority of colonies that we see in the wild are what we call mixed, where some polyps have high densities and some polyps have low densities. And so the fact that they naturally engage in what we would call a facultative symbiosis, right? So they don't require those Symbiodiniaceae partners to survive, gives us the ability to use that naturally manipulated variable to then ask some questions about what's the impact of Symbiodiniaceae density on the microbiome, on who's in the microbiome and what the microbiome could be doing. And this goes back to, sort of broader questions about when corals lose Symbiodiniaceae, what kind of regulation over the microbiome composition are they also losing, right? What are the microbial consequences of coral bleaching? The other thing that's really neat about this symbiosis between Astrangia and its Symbiodiniaceae, and by the way, up at the top of the slide, I had this, I think, in the previous slide too, the name of that alga, the Symbiodiniaceae, it's the genus and species is Breviolum sigmophyllum. And so the other thing that's neat about this symbiosis is that it has an extremely large latitudinal range, actually one of the largest geographic ranges latitudinally of any hard coral so far known. And so that latitudinal range spans over 50 degrees. The populations really are along a wide range of average sea surface temperatures, as well as temperature regimes and light regimes and nutrient regimes. So up here in the northernmost part of the range, and I put a little star, hopefully you can see this, to represent Fort Wetherill, we see, as you all know here in the audience, right? Good New Englanders, we have really, really hot summers and really, really cold winters, right? But down in the tropics, in the Gulf of Mexico, off the coast of Florida, and then a little further north in the subtropics, we really see much more even temperature ranges across the year. So this gives us the opportunity to ask a lot of questions about the influence of seasonality and of these different types of temperature regimes, as well as the influence of the symbiotic state on microbiomes of this coral. And so what I want to do then is, now that I've kind of set up that question for you, is just get right into the first story that we did. And so this was right when I first got to Roger Williams back in 2015, 2016, what we did was we wanted to know just what bacteria, what archaea, what does the microbiome look like in Astrangia poculata in the wild? And so we wanted to know if it was potentially correlated with or directed by symbiont states. You can see there's a white coral right here living directly next to a brown coral in the wild. And then we wanted to know whether or not it was consistent. So what this is a graph of temperatures from a NOAA buoy, average sea surface temperature across a year. So this is January, January all the way out to December. And this was in 2012. And so you can see there's a really large temperature fluctuation, right? On average from about 5 degrees up to the mid 20s, and this is Celsius. And so we wanted to know this basic question: what drives diversity patterns of the microbiome? Um, sorry, in Astrangia? Is it symbiotic state? Is it seasonal variation, or is it both potentially? And so what we did was we went to Fort Wetherill, and by scuba we collected 10 sets of paired colonies. And the way that we defined that was that the 2 colonies had to be within really just a few centimeters of each other. So we knew that they were on a bulk level surrounded by the same seawater. So we could compare the microbiomes, brown 1 with white 1, brown 2, white 2, all the way up through 10 pairs of colonies. And then we also collected seawater from the locations each time that we sampled. And we sampled throughout the seasons, so fall, winter, spring, and summer, for an entire year. And in the summertime, we were lucky that it looked like this, but I have to get a picture. I have a picture somewhere deep in my archives of scuba divers collecting for the spring time point, and during which it was actually snowing. So it depends on the time of year. It's a lovely place to collect sometimes, and other times it is just bitterly cold. So I'll give you the broad sort of 30,000 foot view first of what these microbiomes looked like in profile across different seasons. And really the main take home point is not to try and squint and see, okay, what does red represent? What does orange represent? I want you to look broadly at the patterns. Each color represents basically a type of bacterium actually at the phylum level. So it's a fairly high up, low resolution, you could say, look at the bulk taxonomic diversity of each sample. And what you're looking at here is on the left-hand side of the graph, there are brown colonies, and sort of what it is the average proportion of each of the types of bacterial groups are represented. So each number is actually an average. So all 10 of the brown samples from the fall are represented by this column. Okay, and all 10 of the white colonies from the fall are represented by that column. And so we basically have 4 seasonal mean time points for each of the brown and the white. And so the first thing that we saw, just looking at this on a bulk scale, is that 1, we were thrilled to see that they're very similar in composition to tropical coral microbiomes. So what this means is that we can probably use this to make some comparisons very broadly with coral microbiome dynamics. But another thing that was very interesting is that while they're similar in composition, they're actually quite a bit less diverse. They're quite a bit less complex in terms of the number of phyla represented in tropical corals versus temperate corals. So they're less complex, and actually from one season to the next, there are some significant changes, but they're much more stable than tropical coral microbiomes. They just don't change as much as tropical coral microbiomes. And that's going to become something that's really interesting. Or I'll keep coming back to that. I want to mention also that this was work that was done in collaboration with Frank Stewart's lab at the time Georgia Tech. He's now at Montana State, and with his postdoc, Dr. Zoe Pratt. Now, the other thing that was really exciting for us to see was that this work, which was comparing, or these sequences, I guess, which were comparing brown colony microbiome composition to white colony microbiome composition, they don't really look very different from one another, but they did look quite a bit different from the microbiome composition in seawater that was sampled at each of those time points. One thing that's worth mentioning, I really skipped right over the methods, but the thing that's worth mentioning is that what this is based on is an analysis of the 16S ribosomal RNA gene sequence. So some of you in the crowd might know that 16S is a gene that we use to basically fingerprint different types of bacteria. And so based on the sequences that are in a sample, we can identify and determine what kinds of bacteria and archaea are present in a sample. And so all of these histograms are based on the relative proportion of a particular 16S sequences in each of the samples. And so we see brown and white, even across different, all, on a bulk level, don't really look that different from each other, but they did look different from the seawater. And so that was exciting to see, mainly because it's consistent with what we already know about tropical corals, that they maintain very specific microbiomes that are distinct from those that we see in seawater, even though the brown colonies and the white colonies do not. So symbiotic state did not, at least at this bulk level, seem to be important for, or at least linked to, any differences in microbiome composition. And so another way that we could look at this using statistical analysis was what you're looking at here is a plot that's called an NMDS plot. And all that I want you to think of with this is that the closer two coordinates are to one another, the less dissimilar they are to one another, or the closer they are to one another in terms of microbiome composition. And so what this is indicating is that, so the brown colonies are shown in diamonds as shaded brown diamonds. The white colonies are the white diamonds, and then blue represents seawater sequences. So the first thing that you might see from this is that there's substantial overlap across all three groups of sequences. And again, each one of these points, these coordinates, represents a single sample that was taken. And so there are 10 brown, 10 white, 10 seawaters from each of the 4 seasonal time points. And so there's substantial overlap, but then with statistical analysis, what we were able to determine was that the apoculata microbiomes, brown and white, really are not distinct from one another, but they do represent distinct microbiomes from the seawater. And so this was something that was a real surprise. We did not expect to see that symbiotic state didn't impact the bulk microbiome composition. We thought that if they have these symbionts inside that are photosynthesizing, they're making sugars, those sugars would be in a traditional coral model, what we think is that sugars are released to the mucus. The mucus is where a lot of the microbes in corals tend to be. Why wouldn't it enrich for specific things? And compared to something that doesn't have these things photosynthesizing, that mucus wouldn't be enriched in those things, right? It should be pretty straightforward. So we were really perplexed to see that. But then the story got interesting. And this is what led to sort of like an unexpected left-hand turn in the focus of my research, which was that from one season to the next, we saw dramatic differences. So it turns out that season is a main driver of microbiome structure. So what you're looking at now is the same plots of, this is just the coral colony microbiomes. So the seawater has been removed from this plot, but now they're coded by season. So all of the fall are in this like beautiful scarecrow autumnal orange, right? And then winter is an icy blue. Spring is the this verdant green color that represents what we just went through. And then summer is bright sunny yellow, right? And so, and this work was all done, this analysis was done by another postdoc who we collaborated with, Dr. Allison Kerwin. And what she was able to show with her analysis was that fall was significantly different from winter, significantly different from spring, significantly different from summer. From one season to the next, there were significant shifts in the microbiome. Most interestingly to us, in the spring, do you see how that cluster of green symbols is all much closer together? So what that indicates, that the term that we use when we think about microbiome diversity is low dispersion. In other words, it's very predictable. So as I pick from one colony to another colony of corals, I'm more likely to get very similar microbiomes, as opposed to in the winter or the fall, I'm going to pick from different colonies and potentially get something that's much more different. So it's more predictable. It's more similar to one another during that springtime collection. And it's also what we call beta diversity, right? But so basically lower beta diversity during that springtime. And the thing that was really neat about this, and I won't go into these data today, but that microbiome was dominated by groups of bacteria that we identified as potentially being what we call the core from the microbiome. These were groups of bacteria that were consistent across the entire year. So the idea that we came up with after this was that maybe during these seasonal shifts, from winter to spring particularly, there's something where the bacteria, where the coral or something about the metaorganism itself will shed the things that are not necessary, maintain just the microbial players that are really important for the coral's survival or metabolism. And it's almost, you can almost think of it as sort of like an annual cleansing, an annual reorganization, reshuffling. And so we're really interested in how this could be happening and why this would happen when it does. And then this is the importance. So it was one of the moments where I'll talk about how important it is to talk with other researchers, to collaborate and to be part of a community. I talked with someone who knew a lot about the Astrangia ecology and had been working on Astrangia for a long time. And he introduced me to the concept that, and it had never even occurred to me, corals go into dormancy in the wintertime. These corals do, right? Of course, tropical corals don't because their temperature is always the same throughout the entire year. But in New England, like any smart New Englander, they go into dormancy. And in the wintertime, they go through it. This dormancy is called quiescence. And what they do is the coral polyps themselves will retract. The tentacles go in, the body of the coral goes in, they stop feeding. If we tap them with little metal probes, either out in the wild or in tanks, talk more about that later, they're completely non-responsive. And so what I can tell you right now is that the microbiome during this quiescence, or sort of around this quiescence, I should say, it bears the signatures of tropical corals in what we know as disease. Another term we use for it is dysbiosis, where there's been a disruption of functional biosis, symbiosis. And so that dysbiosis state during or around quiescence, what it was enriched with, or what it was sort of the signatures where that was enriched in these bacterial groups known as Clostridium, Oscillatoria, Rickettsia, things that have been associated with coral disease in the tropics. And it didn't have some of the things that have been known to be important players in coral health in the tropics, Oceanospirillum and Thiohalorhabdales. And so what we were thinking is that quiescence really is corresponding then with a lack of regulation. So it's not an indication that it's been diseased, but that it's lost its regulation of the microbiome of Astrangia. And so there must be something that's going on as it's not feeding, it's not producing things that it normally does, that basically a whole bunch of stuff gets dysregulated. And another way to think about this is that this is an opportunity, and I feel like we sort of stumbled onto this, to study how microbiomes can be reassembled in response to disturbance. How are they organized, and how can they maintain any kind of stability? Right. One thing that I told you a few slides back was that microbiomes in Astrangia were unusually stable compared to tropical corals. That from one season to the next, even though they change a lot less than tropical corals do. So we know that it's consistent, we know that it's stable, and that surrounding this period of dormancy, there appears to be some kind of reorganization to sort of this minimal, important, critical microbiome component. And so one question that we have is that annual disturbance really important for maintaining resilience and stability of the microbiome? And we're now thinking about this seasonal shift every year in the Astrangia physiology as an opportunity to study microbiome recovery from disturbance, and specifically from coral bleaching and from coral disease. And so really the questions right now are what directs the organization of that microbiome? So here's that picture again, showing that decreased dispersion, right? Where really in the springtime, in that time point, we saw that the coral microbiome is dominated by a small subset of microbes. And so this is where one of our current projects sort of why this got started. So we're really interested in studying the microbiology of quiescence to identify microbial players that direct reassembly of the microbiome. And this is work that's being done with Dr. Anya Brown, who's a postdoc, in Dr. Amy April's lab at Woods Hole Oceanographic Institution. And what Anya bravely did last year that I cannot say that I have ever done is she sampled throughout the year, before, during, and after the coldest periods in the seawater. She went out and collected a Strangia in late October. And basically this is before and then during the onset of quiescence. During quiescence in January and February, I cringe even saying these words, and then transitioning back out of quiescence and then sampling after quiescence. So at each of these time points indicated by arrows, she collected corals from the wild. And very quickly, what I'll do is just let you take a look at the visualization of the data that she generated. And so these are again based on 16S profiles. And this animation that you're seeing is going from before quiescence, onset, during, transitioning out of, and then afterward. I'll give you a still here. So basically what she saw was that the community microbiome composition changed through each of these time points significantly. And that a Permanova analysis confirmed that dormancy timing was significantly impacting the coral microbiome composition, and which wasn't really surprising to us. But now what we have is a sort of high resolution data set to understand who the players are that are changing in each one of these steps throughout. So instead of just being a fall sample and a winter sample and a spring sample, we now have a really condensed high resolution time series to start identifying which microbes are key players in this reassembly process. And I won't really share any of those data with you yet because she's still working them up. We're about to submit a manuscript on that project. But what's really been a fun opportunity is that these are some of the undergraduates in my lab. So we have Emma Place and Cassidy Hughes, Marielle McGovern, and then my lab technician, Alicia Shickley, who designed a system in our lab at the Roger Williams Wet Lab to induce quiescence in the laboratory. And over this past last year and this year, what we were able to do was run an experiment where we took corals down into quiescence by chilling them, basically to winter temperatures, and then simulated that period of wintertime quiescence and wintertime temperatures and ramped them down in, held them there, and then ramped them back up, and sampled the microbiome throughout that time series to see whether or not we could simulate quiescence in the laboratory and get similar pictures of microbiome dynamics from that experiment. And so what I can tell you is stay tuned on that. We've got some interesting data coming back from those as well. But this, we hope, will be able to help us answer a lot of questions about the influence of dormancy on microbiome sort of reshuffling and active reassembly. Now, the final microbial vignette is actually very short, but what I want to tell you is that we weren't totally right, we think, about the first thing that we found. So I told you before that we think that symbiotic state didn't really influence microbiome composition, but it turns out that what we now have come up with, and this is through a collaboration with Dr. Amy April as well at Hui and Dr. Shavona Bent, what she was able to find was that when we treated Astrangia colonies in tanks with the stress of antibiotics, right, basically lowering the overall abundance of bacteria in and on the colonies, and then watched them sort of reassemble microbiomes after that disturbance. What we were testing for was whether or not brown colony reassembly looked different than white colony reassembly. And so what I'm showing you here is the experimental setup that she designed. So she basically had 5 brown colonies and 5 white colonies that were treated with antibiotics, and then 5 white and 5 browns that were just in filtered seawater with no treatment. And she sampled the mucus and sequenced 16S genes from each of those samples. She did it right before the treatment at what we call time 0. We do a pre-treatment, a time 0 just after the antibiotic addition, then 96 hours later, a week later, and 2 weeks later. And very quickly, what I can tell you is that what we saw was that, in fact, we think that the browns looked really different than the whites in the sense of how they changed, not necessarily to what state they recovered to, but the process of changing. So if you look at the first panel here, again, this is an NMDS plot that's really similar to what I've been showing you before with the data. All of the sort of orange brown colony, or sorry, coordinates here represent the 5 colonies that were treated with antibiotics and were brown. The yellows represent the white colonies that were treated with antibiotics. And then the blues are the white colonies that were control, and or sort of light blue there. And then the dark blue was brown colonies, not just control, no treatment. And so immediately after the antibiotics, hopefully what you can see here is that there's a split. These ones were treated with antibiotics, these ones were not. They don't look too different. And then they continue throughout the time series to sort of group together. Antibiotics look different than controls. But what was really pretty marked for us was that the size of that ellipse around all the coordinates, what that indicates is basically dispersion, how variable they were among the different colonies of that single treatment. And so it looks like in the brown colonies, it was much tighter, right? So there's something about the white colonies, that lack of, potentially the lack of the symbiont, that sort of leaves it to be much less predictable. Or another way to think about that is there's something that the symbiote is doing to exert influence and what we see as predictability in that response over time. So consistently, they had less inter-individual variability. And what this led us to believe is that maybe that symbiont doesn't influence bulk taxonomic microbiome composition, but it can exert influence over how microbiomes respond to disturbance and how well and how predictably they recover from that disturbance. So we're starting to piece together different parts of the microbiome, or I should say, we're piecing together different influences. What are the things that regulate microbiome composition in this coral Astrangia? And what we think right now is that, as I said, season and especially winter quiescence influences the microbiome composition. It's probably not, what we didn't see was that brown microbiomes look different than white microbiomes. We think that quiescence is really important for shuffling the microbiome, for reassembly of the microbiome. And it's fascinating to think about how that microbiome reassembly could be happening on an annual basis, and the opportunities that that presents in comparing and contrasting this coral to tropical corals, which don't have, we don't have any reason to think that they have an annual reshuffling in that way. And so what we're working on now is characterizing the immediate response to quiescence and sort of surrounding quiescence, both going in and coming back out. And we're doing that both with wild colonies and with laboratory manipulated colonies to see if we can start understanding, piecing together the patterns of diversity and the patterns of response to environmental stress. And after acute disturbance, we have some evidence that the symbiont Breviolum may exert some influence over microbiome recovery, but of course we don't know yet how or why, right? So there's with every question that we've sort of started to answer, we of course have tons of new questions that we want to answer. And these kinds of microbiome studies are, are subjects that really require multidisciplinary approaches. And so I definitely am what my PhD advisor would call a microbial chauvinist. I always think of things in terms of a microbiome perspective. Even when I'm studying animals and when I'm studying ecosystems, I start by thinking about the microbes. But what's wonderful is that if you collaborate and you work with scientific communities, you can work with people who maybe think about the corals first, or maybe think about nitrogen first, or or any sort of other factor that could be involved in determining coral health, coral reef ecosystem health, or even just what is it that controls coral microbiomes, right? And so here's where I just will wrap it up by saying that this was something that was really important to me and to a lot of us who are working on Astrangia, that we had to have a community to move the science of this coral forward. And since I know I'm talking to a large group of undergrads, this is my opportunity to say, this is probably the most important thing that you can do in science, is to collaborate, to talk with each other, to talk with other scientists who, when you first meet them, you might think they do something totally different than what I do, right? But there's a lot of lessons to be learned. It's so funny, Jim, that when we were first talking before I started, that squirrels kept coming up, because actually a lot of hints about what's going on with quiescence microbiomes come from work on squirrels and on how squirrels, endure winter and what happens to squirrel gut microbiomes throughout the year. So there's a lot of different important communication and collaboration that can happen. And so what I want to tell you about really quickly is a community that I was involved in establishing, and why I think that this was so important. And in fact, more than the science, more than any experiment that I ran, to me, this is the thing that of which I am the proudest. And I think it is the most important part of science. So really, students, hear this, that this is where it's at, is working to be part of a community. And if you don't think that community exists, innovate and make that community, because it is possible. We knew that it was really important to have large groups of people able to work with this organism. And this was something that motivated us. And when I say us, I mean Dr. Randy Rojan from Boston University and Dr. Sean Grace from Southern Connecticut State University. The three of us established a working group. It started as just a few people getting together one summer, and it's now grown quite a bit. But we did this because we knew Astrangia was an important organism to work with. We wanted a lot of people to be able to have access to it. One of the pieces that my lab has been involved in is establishing spawning and breeding in captivity of this organism. And so we've now gone through nearly the entire life cycle in captivity. So we can spawn Astrangia in the summer. And we do so in an event now that we've had for several years going every August. We call it Spawn Fest. They predictably spawn in August. And what you can see here is parts of a student they're monitoring colonies that we brought into the lab. Basically, we can collect them from Fort Wetherill, bring them into the lab, put them in still sort of warm water, warmer than what they're in out in the wild. And the result is that they will release eggs or sperm into the water. And so this is a female colony releasing eggs into the water. And what this looks like in real time here. Sometimes we can sort of trigger them to go by just disturbing the water column over them. We're not squirting them with anything, but these are what we call the fireworks. So this is, they're both male and female colonies here in this, this video footage. You can probably see some of the dots. These small white dots in the water are actually eggs. And then some of these other things that look like streams of smoke, like this right here, are sperm being released by male colonies. So we have males and females in here. We can mix them together in the lab and go through fertilization. And so now we've been able to predictably spawn them, fertilize them in the lab, bring them up as larvae. They develop as has been previously described. And just last year, with collaborators with the Smithsonian, we were able to get them to settle onto hard substrate in the lab. And so now this year, we're working to, this August during SpawnFest, we'll be working to repeat that and basically see if we can take them from settling as tiny little, they just look like little dots, into metamorphosed full colonies. And then the super awesome part will be when we raise them up to be old enough that they'll make their own and release their own gametes in the lab. But this, we hope, is something that will support a lot of a large community of researchers. The other thing that we've been able to do is develop using CRISPR-Cas9 technology. We've been able to take embryos and basically microinject them. So what I'm showing you here is basically just microinjection of a fluorescence dye that was injected at the zygote stage. So these are zygotes from Astrangia that are just attached to or sort of immobilized on this microscopic mesh. And that dextran dye then, as the zygote was allowed to continue to go through development, can be shown in just one quadrant of the blastula stage. And that was just to sort of show that we could do microinjection. And this was with, I should say this was led by collaborators Leslie Babonis from Cornell University and Jake Warner from UNC Wilmington. And what they did, they're the dream team who basically created a construct of a gene for something called mini collagen 3 and they connected it with a fluorescent tag, a gene that basically will express a bright fluorescent green um, color when this entire construct is expressed. So this is a gene. And so that gene, what they were able to do was inject it into cells at the embryo stage, let that develop. And then what they found was that in the early larval stages, where we see cells that are developing, that they become basically the stinging cells of the coral, that mini collagen 3 is expressed. And where that mini collagen 3 is expressed, it's expressed with a fluorescent tag. So we see those early stinging cells lighting up in bright green. The other piece of this information that we have is that when we used primers to target this mini collagen 3 gene in what we call wild type, which were basically ones that we didn't manipulate, we could see mini collagen 3 of a known, what the fragment size that we expected to see. The construct, which is indicated by this lettering here, shows that it had that extra tag on the end of it, so it was larger. It came out larger on the gel. And this is what we call a knock-in. We basically knocked that gene into the embryo. Now, we didn't really need to know that this is where mini collagen 3 is made in embryos, but this is a proof of principle that we can do what we call genome editing in this organism. And so now we could come up with all kinds of different genes to query and say, what does this gene do, or where does this gene direct, different developmental processes? How does it impact the onset of symbiosis in corals, right? There are a lot of different things we could now query now that we have this mechanism and this method in place. And so what I'll leave you with is this, is that we've, in the process of sort of developing this small group of people who were really excited about this weird coral, 6 years later, we now have a group of almost 200 scientists who meet annually, although we've just changed it now to every other year. And they're folks who come from all kinds of different backgrounds: symbiosis ecology, microbiome work, physiology, self-other recognition, transcriptomics, genomics, proteomics, metabolomics, reproduction. And so we did miss a year in 2020, but even in 2021, we had really the best virtual conference I've ever attended. It was in gathertown.com. And so this is our group picture of our meeting from 2021. But if you're interested in Astrangia science, or in Oculina science, or the science of temperate corals in general, or if you just want to be with a fantastically collaborative group of scientists, check out these sites to learn more about the group. Contact me if you want to be on the mailing list. We plan to have a 2023 meeting to get all of the temperate coral scientists together to discuss their latest work. And finally, what I would say is that the reason that I've spent so much time working on the science of this coral is that I see it as a great experimental opportunity, as I hope I've convinced you throughout the talk today, but also as an emblem for scientific innovation, an emblem for or sort of an ambassador for our local ecosystems, to get, for example, K through 12 kids out to just know more about what kinds of animals are out in our ecosystems. And the pitch that I made to the state assembly when I first was suggesting that this be the state coral was that I said that it's hardy like New Englanders, it's small like the state of Rhode Island, and it stands to tell us a lot as a local about large global problems, right? And somehow that worked, and that got this established as the official state coral of Rhode Island, the first state coral in the country. And so we're happy to sort of be looking forward to new ideas about how to help incorporate this into STEM education across the state as a state emblem. And so with that, I just want to acknowledge that this is the work I've been talking about today was in collaboration with a lot of people, a large community. And if you look at the list of names in the Sharp Lab, this is all students who at some point or another, including my lab technician, Alicia Shickley, have all been supported by INBRI, by CAIM, and we're very grateful for that, the experiences for these students who have gone through the CAIM program. So with that, I'm happy to take any questions if there's time, and thanks everyone for coming.
Awesome. Thanks, Koty. That was great. Let's give Koty a nice round of applause, and we definitely have some time for questions. So first, some applause. Thank you, Koty. That was awesome. And I definitely have some questions that I'd love to ask, but I see you all the time, so I can hold off and
Great.
let others go first, at least until we run out of questions, and then I'll jump in maybe. But go ahead, guys. Questions? All right.
Hello, I have a question.
Hi.
Is there a specific gene that you're interested in looking into soon with the microinjection sort of method you were talking about?
Oh gosh, that's a good question, Cassidy. It's hard to decide. There are there's a suite of genes that are we think involved in initiating onset of symbiosis with the photosymbiont, with breviolum. There's a lot that's been done in tropical corals in terms of what kinds of sugars in the mucus in tropical corals can sort of entice and help attachment by those photosymbionts. And so those kinds of the things that synthesize those sugars, I think would be right at the top of my list to learn more about what would happen if, what are the analogs in Astrangia in terms of those sort of symbiosis initiators. And whether or not if we manipulate those, if we can see differences in symbiont state. I think that would be my first one. If you have some, please let me know.
Okay,
Happy to
will do. That's
crowdsource
super
that.
cool. One more question, sorry. With the sugars that you're talking about in the mucus, have we looked at if there's a difference in like the amount of those sugars between the white and the brown?
No, I don't believe that is something that's been researched. So back in the early 80s, there was some great work done at URI actually in Mike Pilson's lab. And Mike Pilson is really the sort of the original Astrangia researcher of this century. Astrangia actually has this is a total side note, but Astrangia has this long history of being researched since the 1700s. And some of the first documented research was done in Castle Hill down in Newport. But Mike Pilson's lab is really the research group that brought it into the back into the limelight. And in the early 80s, one of his students did a lot of work on looking at nitrogen composition and the difference between brown and white Astrangia colonies and the types of nitrogen species that were in their mucus layers. But as far as what kinds of sugars are in there, I don't think anybody has done that. We could definitely use some metabolomic folks to help us figure that out or to address that. We'd think there should be differences, right? As I said, but we have not figured that out yet.
Thank you.
Good question.
Any other questions before I go?
I have a question.
Go ahead, Sean.
Hey, Sean.
Hey, how you doing? So you had said that you don't know how breviolum induces recovery out of the quiescent state for Astrangia. Is there any other examples where breviolum does that for maybe a different species, or like, I guess, is there any other species of coral? Maybe it's not breviolum that's inducing recovery, but a different symbiote, or maybe we just don't know enough to answer those questions.
So I honestly, the answer is all of the above. So you're hitting on something that's one of the reasons I'm super excited about this that I didn't get into at all, which is that so breviolum is one of several genera of Symbiodiniaceae that we see in corals across the globe. And so breviolum is a genus of Symbiodiniaceae that is found in tropical corals, but it's found in and I should say it's the dominant type of Symbiodiniaceae in some of the corals that currently are exhibiting the highest level of resilience to a microbial disease. We think it's a microbial disease that's basically ravaging the Caribbean reefs. So there's something called Stony coral tissue loss disease. And what's really interesting is that the corals that seem to be the species that seem to be the most resilient are the ones that typically are dominated by breviolum. So there have been some hypotheses about what breviolum could be doing that could be conferring that resilience. A lot of what we know about these different genera of photosymbionts is that they have different levels of thermal resilience or resilience to probably to disturbance in general. But I have to hypothesize that it may also be some level of interplay between the brev and the microbiome that really hasn't been well described yet. But there's certainly enough data that it's tantalizing to me to think about how could breviolum be, and really just sort of my plan is just to sort of stick with the stranger and say, how is it the breviolum is regulating this response to disturbance? What can we learn from a stranger that can then be translated to understanding what is the control of microbial or microbiome, I guess, resilience or response to environmental disturbance in tropical corals? And that, I mean, that question is underlying all of the work that we do here in Australia, but that's, you hit on one of the big ones.
Koty, related to that, actually, so one of my questions I've had is, what do we know generally about the recruitment of a microbiome? Is it an active process? Because obviously they're changing, and as I go through quiescence and coming out of it, there's something going on. Is it an active like recruitment process? Is it a active like obstructing process? Is it just we don't know?
So we have lots of pieces of the puzzle, but I wouldn't say that we have a really firm, thorough, conclusive model of how it works. We do know we have lots of isolated examples of where there are some bacteria that are really good at, for example, killing known coral pathogens. And we can find those on coral mucus. We know that they're what's sort of a terrible discovery that was made was that they're in fairly high abundance in a specific species of endangered coral in the Keys, Acropora palmata. There are anti-pathogen bacteria in healthy Acropora palmata in pretty high abundance. In August, during these extreme warming events, especially if they're sustained, that bacterial species in particular drops precipitously in abundance. And so we do know that, one of the models is that in response to change, especially increased temperature, some of the most important defensive bacteria disappear, or at least are down functionally to such low levels that they're not able to protect the coral. And so that's one example of what we know some microbes do to regulate the microbiome itself. We also know that just sort of the general microbial ecology, environmental micro theory, is that particular compounds that are present in an environment will enrich for particular types of bacteria. And I always tell my students this in my microbial ecology classes, right? If there's a substance on this planet, there's a microbe that can burn it for fuel, right? That can consume it or break it down. And that's true whether or not we're talking about sugars that are in coral mucus, or we're talking about arsenate, or we're talking about what we know now about plastics, right? There are bacteria that can potentially metabolize those. And when you introduce that substance, you're going to enrich for those types of microbes. By the way, I'm not saying, just side note, I'm not saying that like we're fine and microbes will take care of the plastics problem on the planet. That's not what I meant to intend there. But presence of a substrate will enrich for higher abundance of a particular microbe that can break that down. So it's a combination probably of things that are doing this, like you said, and then other molecules that are doing this, right? And so we don't really know how, like I said, in tropical corals, how Symbiodiniaceae enrich for particular microbes. What we do know is that in non-bleached corals in the tropics, there are pretty distinct microbiomes. And when they bleach, the microbiomes look totally different. And those corals are much more susceptible to invasion by pathogens, right? So it's probably a complicated network of a lot of different interactions, loss of protection, loss of particular things that are enriching for the good guys. It's almost certainly analogous to what happens in human guts. If you think about environmental disturbance rather as a shift in a diet, right, or taking antibiotics, right, it gets rid of the protective stuff. If you take antibiotics, you can potentially get rid of your beneficial microbes, your protective microbes, which allows for overgrowth by other ones that are not necessarily beneficial. If you change your diet from being enriched in protein and fiber to enriched in Cheetos, right? You're going to change the composition of the microbes in your gut. And so it's a little bit of both.
Thank you. All right, we are past time, so I will, unless
Okay.
there are people have questions, I mean, I will hang around for a few minutes. People have anything they need from me. I don't know, Cody, if you want to just hang around for a
I'm
couple
happy to
minutes.
hang around if anybody has any
But
questions.
I want to thank everybody for joining us, and I hope to see you guys next week at our seminars. And so with that, we'll give Cody one more round of applause, and then those who need to leave can leave, and those who want to stay and talk, they're happy to stay and chat for a bit. Thank you, guys.
Thanks, everybody.
Bye.
Thank you.
Good, that was great.
Thank you.
Thank you.
I have another question.
Yeah.
Because one of your early slides, you had sort of the dispersion of the coral from the north to the south on the East
Mhm.
Coast. I think this is just a pure curiosity in my brain, because even though I'm fascinated that Astrangia pops up in the north and also in the south along the eastern seaboard, I'm also curious as to why it doesn't show up in, say, like the Gulf of Maine. Or is that just because we haven't studied or looked for it there yet? Or is there something, because I think of the Gulf of Maine and Narragansett Bay as being maybe not the same, but
Mhm.
similar underwater ecosystems. I could be wrong. I mean, I'm not a microbiologist, but and to go a gap from Narragansett Bay to where is that off of southern New Jersey? What's happening in between there that's making it so strange it doesn't show?
Um, actually, that's probably not. It's not really accurate. We definitely see it throughout the Long Island Sound and up the coast of Jersey. So there probably just aren't there just aren't enough dots there. But so your first question though, why don't we see them as far north as the Gulf of Maine? I love that I have like a real answer for this, which is that we did a model for that. We modeled it, and basically the northernmost range right now, and we actually did some scouting, and a lot of this, a lot of this was Randy Rogen and Dan Thornhill, back in the like 2004 to 2008 timeframe. They looked up in the Gulf of Maine. They've looked for Astrangia, and have not been able to find it. And what we determined actually in Diamond et al. 2012 was that the top of the range is determined by the thermal limit of Astrangia. So, I kind of built it up as this coral that's really good at going through quiescence and being able to withstand these cold winters. But what I didn't mention is that oftentimes what we see when we follow the colonies, and researchers have documented this, is that throughout the winters, a lot of colonies will actually experience polyp loss. And there is a cost to going through the winter. And I think going too far north, the cost is too great. And so that's the thing that limits their range, is their ability to grow in the summer enough to be able to afford to stick around in the winter. And then it's kind of the opposite down in the southernmost range. We see them very, very, actually Jamestown is probably one of the most vibrant places for Astrangia populations. It's and Narragansett Bay, I should say, in general. When we see them, we see these walls that are just covered with them. Now down in Florida, where Astrangia has been documented, it's competing with other corals that thrive on sunlight rather than eating, right? And it's in these like super, food-free waters. So there's not a lot to eat. And so it's much more well suited for, sort of that sweet spot where it's cold, but not too cold. And it's full of nutrients. And so that's why we think that's what determines the range of Astrangia. It's weird. It's a very different coral than.
Is it found on the coast of England and out that way?
So it has been documented on the west coast of Africa across.
Okay.
I'm trying to think of. I don't actually know where, but along the western coast of the African continent, I know that it has been documented. It's been found in the UK as a species that rafted up there. So it was actually found documented on a piece of styrofoam that washed up in.
It seems that the Gulf Coast on the Gulf Stream would
Yes.
just bring it there.
Yeah,
Like,
and periodically people will write these little reports of we found Astrangia on a turtle carapace or on, some other substrate and in a weird place, but alive, right? And if you ask Mike Pilson, he'll tell you that he's been able to keep Astrangia colonies alive in like a beaker of seawater just sitting on his desk for months or years, and periodically he'd drop a piece of hamburger in or something, right? There are these sort of legendary stories about how robust Astrangia is. But yeah, they definitely have made it across the ocean before.
They haven't established, like.
I don't think so. I don't think they're actively growing. I don't think that's been documented in the UK. But I think further south, I think down in the lower latitudes, yes.
Okay.
So, and then what's really neat is that we know that there are other species of Astrangia out on the west coast of the US. And so our collaborator, Anya Brown, who's leaving, I just told you was leaving for Bodega Bay, is going to make that her new mission to find another species of Astrangia out there in California.
Yeah, perfect.
Yeah, I was going to ask, like, are there other species? Because I'd be curious to see other coral species living in colder waters that are going through periods of quiescence and what their microbiomes look like and how they're recovering when the water gets warmer. I don't know how many different coral species there are in colder waters, but I'd be curious to see that.
Well, so there are a lot of cold water corals, like the corals that are found down deep, right, in these sort of deep reef systems, but they stay cold year round, right? They're just sitting at about 8 degrees all the time. And so they don't work for these quiescence questions. There's another species of temperate coral along the east coast of the US, that's Oculina is the genus. And but Oculina only goes as far north as North Carolina. We've never looked for it, but my guess is that they don't go through quiescence. And we also expect that Astrangia poculata that's that far south also doesn't go through quiescence. It is one of the, well, actually I have a couple proposals in about this right now, but comparing populations from the south to populations from the north and looking at microbiome, seasonal changes in microbiomes and sort of overall predictability and diversity patterns in ones that probably don't go through quiescence versus ones that do. I think that's going to allow us to sort of fill in the gaps on some of these questions that we've been having, is how does that annual reset impact the overall composition and stability of the microbiome? It's super exciting. I mean, this is why I love working on Astrangia, is that there's always another question to come up with.
I'll have to write a story on this again in the fall. It's been a while since I've written anything on Astrangia.
Oh,
but
we'd love that.
Yeah.
We'd love that. You should come. And Sean, we also I don't know if I've told you or if anybody else has told you, but we just built a new, or sort of renovated a new space in our building.
Okay.
And
yeah.
so we have like a super pretty molecular lab now. It's a great opportunity for good photogenic spot.
Yeah.
Yeah,
right.
absolutely.
All right.
Awesome.
Come visit us.
Yeah, thank you.
All right. Well, thanks, Koty.
Thank you, Jim. That was so fun.
Yeah, it's always fun, right? I appreciate that. It's always great. It's funny too, like as much as we chat on a regular basis, like I feel like I don't know what's going on, like in your sort of research world, right? So
right.
I know, but I don't
seminars.
know.
We need these seminars. They're so good I mean, I wish I could just we could come up with a way to just have a day where everybody in our building just gives a talk. And so I know more about we see each other all the time, but.
Right. And so just, I guess we take it for granted in some ways, what's going on. But
it'll go up on YouTube as well. So I'll put it up on social media probably like next week or something like that.
Sweet.
Which I can stop recording.
Thank you.